The next topic is Human Genome Project. What is meant by human genome? Genome means a complete copy of entire genetic information present in a cell. In human beings, this genetic information is distributed in 22 autosomes and 2 allosomes, X and Y, altogether 24 chromosomes. It is distributed in total 24 chromosomes. The genetic makeup of an organism or an individual lies in the DNA sequence. DNA is the blueprint of life, and all aspects of an organism is in the form of a coded information in the DNA. If two individuals differ, then their DNA sequence should also be different, at least at some points. That is, no two individuals' DNA is alike. At least at certain areas, there is some difference. Human Genome Project was closely associated with rapid development of a new area in biology, that is bioinformatics. This is a new field in biology, bioinformatics. Now, goals of Human Genome Project. The first one is to identify all the genes, that is approximately 20 to 25,000 genes present in the human cells, human DNA. Then determine the sequence of 3 billion chemical base pairs, that is nitrogen base pairs. We have to determine all these 3 billions of nitrogen base pairs present in the DNA. Then we have to store this information in database. For this, we have to generate special, computers, that the programmed computers. Later, to improve the tools for data analysis, simply by sequencing DNA is of no use, and you have to analyze these sequences. For this, you have to develop a new tools. Later, transfer these technologies to other sectors, more particularly industries, for application purpose, so that the scientific knowledge is transferred to the industrial sector, and so that we will get various products, especially which are involved in the medicine. Then finally, address the ethical, legal and social issues, LC, that may arise from the project. How one scientific research, it creates ethical, legal and social problems. This is very interesting. For example, during this project, scientists, they collected DNA samples from various parts of country as well world. In one remote village of Haryana, they collected the human DNA samples, and for their astonishment, their DNAs are exactly resembling the Macedonian people's DNA. An Indian with Macedonian DNA. How this is possible? Actually, at the time of invasion of India by Alexander, some of his soldiers, they retained here in our country. They didn't return to the Macedonia, and their descendants still there in this remote village of Haryana. Definitely it causes certain social issues. If you reveal this secret to the public, they start teaching these people that you are not Indians and go back to your Macedonia. It creates unnecessary social problems, and the scientific committee must address this type of issues, LC. Right. Then significance. Knowledge about the effects of DNA variations can lead to revolutionary new ways to diagnose, treatment, and someday prevent the thousands of disorders, more particularly the genetic disorders. This is one great advantage. Then investigation of non-human organisms, DNA sequences can lead to an understanding of their natural capabilities that can apply solving challenges in healthcare, agriculture, energy production and environmental remediation. Actually, along with Human Genome Project, scientists also sequenced various organisms' DNA like bacteria, E. coli, and other microorganisms like Escherichia coli and yeast, Saccharomyces, plants like Arabidopsis thaliana and rice, Oryza sativa, and other primates like chimpanzee, gorilla, etc. For what purpose? To understand these organisms' natural capabilities, which may help in solving various challenges in our healthcare, agriculture, energy production, environmental remediation. For example, energy production. Nowadays, these ethane and methane gases also used as fuels in certain areas. By genetically, say, engineering microorganisms, we can produce these, say, energy sources in large quantities. Similarly, environmental remediation, that is, they provide solution for pollution control by using certain microorganisms. So these are the various advantages of this DNA project. Many non-human organisms such as bacteria, yeast, then Caenorhabditis elegans, Drosophila, rice and Arabidopsis, these are the plants, etc., have also been sequenced for especially to understand the evolutionary aspects. These are the some important aspects. Now come to the methodologies. The methods involve two major approaches. ESTs, expressed sequence tags, first method, and second method is that is the sequence annotation. It's a blind approach. What are these two? We'll see. Then for sequencing, the total DNA from a single cell is isolated and converted into random fragments, which are later on used for sequencing by using an automated sequencer, which works on basic principle and generated by or invented by Frederick Sanger. In methodology, we need certain host as well vectors. The hosts are generally bacteria and yeast cells, and vectors are bacterial artificial chromosome and yeast artificial chromosome. These are the vectors, and hosts are bacteria as well yeast. The fragments are sequenced using automated DNA sequencer. That is, this is not humanly possible with the help of an automated DNA sequencer, which works on the principle developed by Frederick Sanger. And he is the man who also invented the amino acid sequence of a polypeptide chain. These sequences were then arranged based on some overlapping regions present in them. This required generation of overlapping fragments for sequencing. That is, before we had to generate overlapping fragments by using different kinds of restriction enzymes. Alignment of these sequences was humanly not possible. Therefore, specialized computer-based programs were developed. So, with the help of a computer program, we can arrange all these in the correct sequence. These sequences were subsequently annotated and were assigned to each chromosome. Annotation means adding an explanatory note. And we had to assign these sequences to a particular region of the chromosome. Now see there's two approaches. First approach is the EST, express sequence tags. What is this technique? See, this is a DNA. These are some genes. Gene A, gene B, gene C, like this. Expression means conversion of chemical information present in a gene into an useful product for a cell. For example, take this gene B. For its expression, first off, it has to undergo transcription. As a result of this, one mRNA molecule is formed. This mRNA molecule later on undergoes translation, forming a polypeptide chain. This is the product. So there are two steps are there: transcription, translation. In the EST, express sequence tags, first of all, we had to collect this mRNA from the cell. Based on this mRNA, we had to produce a complementary DNA, cDNA. This is the complementary DNA, cDNA. By using autoradiography, that is, we had to tag one radioactive isotope to this cDNA and leave this cDNA tagged with radioactive isotope onto the DNA. This cDNA, it goes and hybridizes the DNA exactly at this location only. So we can identify this is the gene that codes for this polypeptide. So like this, we can identify the gene. Later, we have to cut this DNA sequence, DNA piece, and we have to use this for DNA sequencing. In the express sequence tag method, we can identify and sequence only the genes that were already expressed, and we can't identify the genes that are not expressed. So this is one advantage and one shortcome. Express sequence tags are very much useful to identify a gene in the genome with 100% accuracy. But we can't identify unexpressed genes, a shortcome of this technique. Second method, in this second method, sequence annotation method. In this method, we have to sequence the entire genome. That is coding sequences, non-coding sequence, everything. Later, we have to assign their functions to this DNA molecule. For example, after sequencing, we have to assign their function. Suppose take this segment, whether this segment is having any coded information or not. If it is having some coded information, again, we have to go for further research, whether it is a structural gene or functional gene, whether it is a promoter or regulator, or whether it is an initiating codon or terminating codon, like this. So we have to assign this sequence with their functions. This is called sequence annotation. Annotation means adding a note on these particular regions. So these are the two basic approaches. Whether it is express sequence tag technique or sequence annotation. Finally, we have to go for sequencing. Sequencing. This is actually a very difficult process, a daunting process, where we need, suppose this is the stretch of DNA to be sequenced. As DNA is a very long polymer, we can't sequence this entire stretch of DNA at a time. We have to cut this DNA into certain fragments. Say here, like fragment A, B, C, D, and so on, like this. After fragmentation, each fragment is sent to a corresponding laboratory for sequencing. For example, fragment A, it contains 1000 base pairs or 1 kilo base pair. How many such fragments are needed to identify all these 1000 base pairs? Actually, we need 1000 copies, because one fragment of DNA is needed to identify one nitrogen base. For 1000 nitrogen bases, we need 1000 such copies. For this, we need certain vectors and hosts. We need certain vectors and hosts. Vectors. The commonly used vectors are BAC and YAC. BAC, bacterial artificial chromosome, YAC, yeast artificial chromosome. For example, bacterial artificial chromosome, this is, first of all, we have to insert this fragment into this BAC, like this. Now we got one recombinant DNA. Later, we have to transfer this rDNA into a convenient host like E. coli, is the Escherichia coli, or even into yeast. Suppose if we transfer this rDNA into E. coli, in the laboratory, if we provide suitable conditions for replication of DNA, this yeast cell, this Escherichia coli, undergoes rapid binary fission. That is, 1 becomes 2, 2 becomes 4, 4 becomes 8, and so on. During this, along with the bacterial cell, the recombinant DNA also multiplies like this. So we got required number of fragment A's, thousands together. This is called gene cloning. By using these multiplied fragments of A, we can complete the sequence. So this is what you call sequencing of a small fragment of DNA. But how many fragments are there? Not one or two, many. And each laboratory is succeeded in completing the sequencing of all these fragments. But next problem is we have to arrange these fragments into correct order. Arranging these fragments of DNA into correct order is not humanly possible. This is actually by using certain computers. And moreover, this arrangement of these fragments based on the overlapping sequences. Overlapping sequence means, suppose this is the DNA. By using different restriction enzymes, we have to cut the same DNA molecules at two different sites. We will get two different DNA fragments. One fragment is like this, one more fragment is like this, and these are the overlapping fragments. A computer recognizes these overlapping fragments and arrange them in the correct order, A, B, then D, like this. So this arrangement is possible only based on the overlapping sequences, right? So this is the DNA sequencing method. Next, another challenging task was assigning the genetic and physical maps on the genome. This was generated using information on polymorphism of restriction endonuclease recognized sites and some repetitive DNA sequences known as microsatellites. Right. What is this? Another challenging task is generation of genetic maps and physical maps. Genetic map, it gives you only the relative position of genes on chromosome, like this. Suppose this is gene A, gene B, gene C, gene D, like this. This can be generated by using percent recombination, that the crossover percentage. For example, the distance between A and B, say 100 centimorgans. Distance between B and C, say 20 centimorgans. Then the distance between gene A and C altogether 120 centimorgans. This type of generation of maps is called genetic maps. But genetic maps gives you only the relative distances between different genes, but not the exact position of genes. Then physical maps. The advantage of this physical map is where we can pinpoint the position of a gene. For example, take this gene A. After identification of gene A, we had to cut this region of DNA into multiple fragments, as I told you previously, and you had to sequence each fragment separately, and finally arranging them according to their overlapping sequences so that you can reconstruct the entire sequence of the gene. So this is what you call physical mapping. In physical mapping, with 100% accuracy, we can identify a gene on the DNA and we have to sequence it. Arrange the fragments according to the overlapping fragments. This is what you call physical mapping. This physical mapping is possible based on the polymorphism in the restriction enzyme recognition sites, as well based on the microsatellites. Microsatellites are one kind of repetitive sequences which also useful in the construction of physical maps. So these are various techniques used in the generation of or sequencing of the DNA. Next, Silent features of human genome. The human genome contains 3164.7 million nucleotide bases. Human genome contains 3164.7 million nucleotide bases. It's a very large number. The average gene consists of 3000 base pairs, but the size varies greatly from gene to gene. The largest gene is the dystrophin gene with 2.4 million bases. This is the largest gene, dystrophin gene. The total number of genes is estimated at 30,000. That is very much lower than the previous estimates of 80,000 to 1,40,000 genes. Actually, scientists before starting this project, we human beings as highly evolved species, they expected very large number of genes between 80,000 to 1,40,000. But finally, the number was estimated to be only just 30,000. Very less number. And almost 99% of nucleotide sequences are exactly same in all individuals. The difference is only 0.1%. And what is the significance of difference, how it is possible, we see in the next session. The functions are known for only over 50% of discovered genes. That is, remaining 50% of genes function is not known to us. Less than 2% of genome codes for proteins. That is, most of the DNA sequence is not coding sequences, only just 2%. Repeated sequences make up a very large portion of the human genome. They are thought to have no direct coding functions, but they shed light on chromosome structure, dynamics, and evolution. Actually, our genome contains nearly 98% of genome is not actual coding significance. It is only non-coding portion. Is there any significance of this non-coding portion? Yes. They shed light on chromosome structure. For example, centromere, telomeres, they are very special regions of chromosome. But here, the DNA is non-coding, yet having some structural significance. Centromere, during cell division, the mitotic spindle or spindle fiber, they attach to centromere only. Similarly, the telomeres, the tips of chromosomes, they maintain polarity of the chromosomes, preventing chromosomal mutations. That is, the DNA at these two regions are having some significance structurally. And dynamics, that is the behavior of chromosomes during cell division, as well evolution. For example, why our genome contains a large non-coding protein, non-coding sequences? It shed light on human evolution actually. Mammals were evolved from reptiles. That is, our genome still contains so many reptilian genes, but these genes are already switched off. Reptiles were evolved from, that is, the amphibians. Our genome also still contains certain amphibian genes which are switched off. Amphibians are evolved from fishes. That is, our genome also contains certain, say, genes related to Pisces. But all these genes are switched off, providing a striking proof for evolution. During course of evolution, we have accumulated all these genes, but majority of them, they turn into non-functional. As human beings are mammals, only 2% of genome is useful for us. That's why a majority of human genome contains non-coding sequences. Then chromosome 1 has maximum number of genes of 2968. Of these 2968, for example, RH factor genes are present on the first chromosome. Y chromosome has very little number, less number, 231 genes. For example, testicular determining factor, this gene is also present on Y chromosome. Scientists have identified about 1.4 million locations where single base DNA differences or SNPs, that is, single nucleotide polymorphism, are present in 1.4 loci. What is a SNP? Suppose this is a pair of homologous chromosomes, like this. At this locus, if you see the DNA sequence, it is like this. something like this. If you take the DNA from same locus from one more different person, it also almost like this. If you compare this, the difference is only at one nitrogen base. Instead of C, it is A. This type of polymorphism is called SNP, single nucleotide base pair differences. There are 1.4 million loci are identified in our genome. What is the significance? These SNPs are present in between the genes. Suppose this is a SNP. The significance of this SNP is it affects the functioning of nearby gene so that the gene may not function properly. If a gene is not functioning properly, it results in certain genetic disorders, diseases. This is what you call disease association. The significance of SNPs is to recognize disease association if you observe the family history of certain families. And so that by using gene therapy, we can rectify this defect in future. This is the advantage. Now, applications and future challenges. Deriving meaningful knowledge from the DNA sequences will leading to our understanding of biological systems. That is, to develop our knowledge in biology, especially during cell division, cell differentiation, growth, and gene regulation, gene functioning, and coordination of different genes, we need this much of information. Second one of the greatest impacts of having this human genome sequence is that one can study all the genes in a genome in a particular tissue or organ or tumor. Previously, it is not possible like this. Scientists, they were able to study only one single gene or a very few number of genes at a time. But after completion of this project, now we are having a chance to study all the genes in a complete organism or all transcripts of an organ. We can study at a time, and you can also understand the gene interaction in better manner. So these are the advantages of application of this Human Genome Project in the biology.